on pregnancy and development. Pregnancy is a common term, but the definition strictly means all the events that occur from fertilization until the baby's born. So I may use some other terms you're unfamiliar with. Conceptus means the developing offspring. Gestation period kind of means the same thing as pregnancy, but it also includes that time from the last menstrual period until birth. Remember I said last time you kind of add 2 weeks. We'll talk mostly about these two stages. We spent a lot of time talking about from fertilization to 2 weeks, the first 2 weeks of pregnancy. We'll spend a little bit of time talking about 3rd to 8 weeks. I'll spend most of the time talking about how the placenta works. We don't really talk about organogenesis in fetal development, which is technically 9th week through birth. But I will talk about some of the things that are done to give birth, like a cervical exam. So development in one slide is shown here. Fertilization accomplishes one cell, and which obviously grows all the way to a full-grown fetus or baby. Again, we'll spend most of the time talking about these early developmental things. If you look at our models over there, we have a lot of models talking about embryology development. And but let's start actually prior to fertilization. If you look at this picture of the female anatomy, the ejaculate deposited in the vaginal fornix, say right here, is something like 300 million sperm. Okay. So they have to swim up through the external os, cervical canal, internal os, all the way up here, all the way through there. By the time you get to here, maybe you're down to 300,000. And if there's an oocyte waiting to be fertilized in the ampulla of the uterine tube, maybe by the time sperm get there, you're down to hundreds, or maybe 100, something like that. Okay, so there's a remarkable attrition from 300 mil to there. The female reproductive tract is a harsh environment for cells, so you lose a lot. The distance from here to here is only about 5 inches, but that's a lot of, 5 inches is quite a bit for cells to travel. So when you get to there, as the sperm kind of swim up through the female reproductive tract, there are things that have to happen so one sperm can fertilize, and we call those pre-fertilization events. So prior to fertilization, things must happen. The most important thing is ovulation. The oocyte is viable in the oviduct uterine tube for about a day. Okay. Now, sperm could be viable for a few days. So for fertilization to occur, technically, ejaculation must occur no more than 3 days prior to ovulation or 24 hours after. So they give you like a 4-day window, but really the best day to fertilize is the day of ovulation, that one day. So fertilization is when the sperm fuses with an oocyte to form a zygote. And I have a series of slides that show you the steps of fertilization. But again, I'm saying things need to happen prior, like ovulation, but also for the sperm, pre-fertilization. So you must have ovulation, but you also must have what's called sperm capacitation. basically that's a softening of the sperm head. And that occurs inside the female reproductive tract, okay? Because of that harsh acidic environment of the vagina, and the other places the sperm is swimming through. So the harsh acidic environment of the female reproductive tract. It's like it kind of sloughs off the harder outer shell of the head, the layers, if you will. Sheds outer layers of sperm head. Like I said, you kind of soften it in that way. And this is kind of like the final maturation of sperm. And it has to happen inside the female, okay, in the ejaculate. And the reason for this is included in pre-fertilization events is because, if you recall, the sperm head has that acrosome over the nuclear DNA, which is the haploid material. There's the nuke in red, right? And the goal is to soften the head so that that double membrane acrosome with its lytic enzymes inside these vesicles will spill onto the egg, the oocyte, okay? So acrosome in green, sperm nuke in red, containing 23 single chromosomes, and use a different color for the plasma membrane. So that's just the sperm head. We went over the parts last time. Cell membrane, plasma membrane. The goal is to have an acrosomal reaction, which is a term on there, which is a fusion between the plasma membrane and this outer layer of the acrosome. That fusion will allow the lytic vesicles to spill and digest the barriers that surround the oocyte. Okay? Write that down. So sperm capacitation kind of precedes acrosomal reaction. Spill lytic chemicals onto barriers surrounding oocyte. So even if a sperm gets there too fast, can't do anything. You got to wait for this sperm capacitation. So it'll get there, it just has to wait until sperm capacitation softens it enough so you can have it have an acrosomal reaction. And you don't want to have the acrosomal reaction too soon, far away from the egg. Then all the sperm will just auto-digest themselves. You want to digest the barriers around the oocyte. Right? And it's not like the first one's going to be the one to fertilize. The first wave of sperm will spill these lytic chemicals, punching holes in the barriers, and maybe in the second wave, one will fertilize. So here's a picture of well, I just drew it there. AC is acrosomal cap, PM is plasma membrane. And you can see it's like many layers. It's hard to see, but it's like there's flaky layers, and you have to like slough them off and just swim along. Anyways, the sperm nuke, the dark material, has the chromosomes. And here are the barriers that I'm referring to. Picture does a good job showing you. This is what is ovulated into the oocyte. So inside there is the secondary oocyte. Look how it's stuck in anaphase 2. It's awaiting for a sperm to get there so it can finish. So the secondary oocyte is surrounded by, going from outer to inner, corona radiata, that means, corona means crown, like a radiant crown. I mean, really, it's pointing to cells that were from the Graafian follicles. So these are granulosa cells. It's like when you ovulated the oocyte, you took some of the follicle cells with it. Okay? And they have kind of a protective function, and there's some cell-to-cell communication saying, I don't know exactly the molecular biology, but basically the cells are communicating, stop meiosis 2, freeze at anaphase 2, right? Stop. Okay. But anyways, that barrier are granulosa cells. The other barrier is the zona pellucida. Yeah, I'll describe it as a glycoprotein eggshell. So don't think literal eggshell, but it's kind of like one. Importantly, there's proteins, ZP proteins, 1 through 4. ZP3 is pretty important. I don't think your book mentions ZP3. I'll throw it up here. ZP3 will bind sperm. It's species specific. So dog sperm can't fertilize a human egg. I mean, there's specificity there. Species specific. So that's important. Okay. Now the goal is to kind of like weave your way through both barriers, as the figure is kind of showing you there. Here's the figure from the Marriott book. It takes you through several steps, and they kind of call this the approach, right? You haven't fertilized yet, so this is all under the guise of pre-fertilization. Until sperm nuke gets inside, then you start to have what's called fertilization. So call this like pre-fertilization sperm penetration. That's kind of what the book will call it. Still pre. So they show you kind of a piece of the oocyte. So it's like, so you got, let's call this the secondary oocyte cell membrane. It's stuck in anaphase 2, sperm's getting there, and it won't complete, but notice that in the outer edge, in the cortex of this cell, there's all these little granules, cortical granules. I'll draw as little blue balls. These are important, these cortical granules. Cortical meaning they're not on the center of the cell, they're on the outer edge of the cell, close to the cell membrane, ready for secretion. Cortical granules. Because you want them good to go. Because as soon as one sperm gets in, you want these granules to like burst out. What they'll do is they'll harden ZP to keep all the other sperm out. If 2 sperm get in there, no go. Never gonna get past one cell stage. Right? So it's like they're good to go. They're out there waiting to be released. But you gotta wait for the sperm to get there first to trigger a cellular reaction. Okay, well, in the cell membrane, there's all these little sperm receptors. So the sperm receptor is basically, well, they find sperm, okay? I think that's obvious. So that you have a second level of species specificity. One is ZP3, the second is the actual sperm receptors in the oocyte plasma membrane. Then they draw a little gap, and then you have zona pellucida itself. I'll just put ZP, right? I've been using that as an abbreviation for zona pellucida. But it's got all of those proteins in it. They draw these little balls there. Okay, the one I mentioned was ZP3. I'll draw some orange balls there. So orange is ZP3. And then on top of that, the zona pellucida, the second barrier was a few layers of granulosa cells. I'll just kind of draw some cuboidal cells here. Few layers. Granulosa cells. I mean, literally we call it corona radiata, crown of cells. So the first thing is, they call this approach. You must breach granulosa cells of corona radiata. So I'll read what it has in the slide there. I know it's hard to read. Very small print. That says approach. Aided by enzymes on its surface, a sperm cell weaves its way past corona radiata. There's enzymes that can break the cell connections between the cells or the granulosa cells, so it can at least breach the first barrier. Approach. Step 1. Enzymes, I'll paraphrase. Enzymes on sperm head. How it breach corona radiata. So you're going to make it to the next layer or barrier, zona pellucida. So remember, you got the sperm nucleus, little N in there. It's got the green cap, which is the actosome that surrounds the plasma membrane. So the next step, looking at the left, they say acrosomal reaction. I'll read what it says here. The binding of sperm to receptors in the zona pellucida causes calcium levels within the sperm to rise, triggering acrosomal reaction. Acrosomal enzymes from many sperm digest holes through zona pellucida, clearing the path to the oocyte membrane. So that's what's shown there. So acrosomal reaction is step 2. Okay, this is the part where I said species specificity. You bind ZP3. So I'll just write sperm binds ZP3, triggering this reaction. So what that looks like for this, the outer layer of the acrosome and the plasma membrane, they're going to come together and they're going to fuse. So that might look like, so we got sperm nuke in the middle here. We got this outer layer that fuses with the inner layer. So after it's all done, you got that inner layer still. And it may be kind of flapped out like that because it's fused with the outer layer. So maybe the plasma membrane is like all fused with it. Blue is still plasma membrane in my illustration here. But you got the inner membrane and all the lytic chemicals have been released. All these little vesicles have released onto ZP to digest it. So I'm writing digest ZP. Okay, the zona pellucida. And so that's the acrosomal reaction there. So you go from here to there. And on the figure, they kind of like show that little red cloud to illustrate you're eating through. So bind ZP3, little orange ball. You trigger this reaction, and you just start to like eat your way through. So that's exactly what happens in the third step. That allows a sperm to get in, and they call it binding. The sperm's membrane binds to the oocyte's sperm binding receptors. Step 3. Okay, so I'll erase this now. Sperm binds oocytes, sperm binding receptors. I can't animate this, but to kind of drill through the fertilizing sperm, there's this kind of corkscrew thing with a tail kind of a spiral drill action. So you can kind of breach all the way past ZP and get to here. So I'm going to draw the head kind of like tilted on the side, the neck will kind of pivot, so you have more surface area onto the sperm binding receptor there. Okay. So we're getting there. The binding receptor, then you have fusion. Sperm and oocyte plasma membranes fuse. Step 4. Fusion of cell membranes between sperm and oocyte. So when they become one, more or less, I'm going to leave a gap in my little sperm tail there, because that'll allow the nucleus that's inside. So we got the nucleus in there. I'll draw it in red, as I've always done. That's going to allow the sperm genetic material to get in, okay, when you fuse. So that's the last step. 5, you let the sperm nucleus in, and it'll block the polyspermy. So it says oocyte sperm binding membrane receptors, okay, oocyte sperm binding membrane receptors are shed. Calcium levels in the oocyte cytoplasm rise. That triggers a cortical reaction. That's the exocytosis of the cortical granules. As a result, zona pellucida hardens, and the zona pellucida's sperm binding receptors are clipped off. That's what's shown there. Sperm nuke got in, cortical granules release. So let's call this the cortical reaction. Cortical reaction by the oocyte is a block to polyspermy. It's a block to letting more than one sperm in. It's a block to polyspermy. So the block is the cortical granules harden zona pellucida, and that kind of clips all the other sperm off. Cortical granules. So I just drew an arrow from cortical granules to my paragraph. Help to harden ZP, zona pellucida, to clip off other sperm. They're all trying to get in, but the first one is kind of like got in. This happens. So according to my picture, basically you just kind of let the sperm nuke in. That's it. That's all that gets in. Nothing else. No tail, nothing else. That's the only contribution is the genetic material. And then the cortical reaction is all of these corticals, they're all being exocytosed. Let me just kind of draw all this in blue. And what that's doing, it's hardening zona pellucida. So I'll just draw these lines to illustrate it's being fortified. Too difficult to breach. So if only one got in, that pretty much ends pre-fertilization. You can actually talk about fertilization now. the genetic material got in. Here's a picture of a model we have in the room right over there. We illustrated the little tail there. But anyways, that's the sperm trying to get in. And the barriers are both shown. You can see the cells are corona radiata, granulosa cells. That would be what number one is trying to tell you. Number two, well, what's number two then? Identify number two. Zona pellucida. So number three is the oocyte. Number four will be just the nucleus of the ovum, the oocyte. On that model, pretty easy to understand. Here's kind of like the same figure. That's an illustration from your book. So I'm going to erase all this now, but we're picking up right where we left off. Now you have events of fertilization. The sperm nuke got in, and they show you all the other things that are going on. So as we go through these figures, look at the barriers. Both barriers are still there. The corona radiata and zona pellucida, they're still there at the start of fertilization. Eventually they have to go away, but they're still there. So these are the events of fertilization. I'll just say sperm nuke got in, but look at, you take an inventory of everything else you see. Check mark, corona radiata still there. You know, ZP, zona pellucida, check the box, it's still there. And if it's still there, you should see the polar body stuck outside the oocyte, inside the zona pellucida. There's the first one. It's going through a meiotic division. It will become two polar bodies, but that's the first polar body. It's stuck inside ZP. First polar body. Check, it's there. Okay, and what it's showing you is anaphase 2 is going to complete. I'll just say will proceed. It was kind of stuck there until sperm got there, now it's there, it can proceed. Now, we have some models in the room that kind of approximate this part of fertilization. Here's what we just looked at. I see two nuclei here. Now, what do you think that belongs to, sperm or oocyte? I would guess belongs to that, sperm. This sperm got in. This sperm now can get in. Yeah, that's the fertilizing sperm. So these two need to come together. I kind of call this like, that's N, right? That's N2. I kind of call this like N plus N.
So the first N is the chromosomes from the sperm?
Yes.
Okay.
So it's like, okay, this is from sperm, drawn in blue. I'll call that an N.
There we go.
And then, I don't know, you have another one from the mom. You can call that N plus N. Because they haven't quite fused yet. But they're in one cell. Now, you won't see that in the book. That's just kind of my thing. That's how I think of it. This is prior to the fusion of the genetic material. They haven't intermixed yet. So, for example, on this model, I don't see corona radiata. It's gone. But I do see zona pellucida. So that's number one. Number two is the oocyte. So number three is the nucleus. Number four, they show the chromosomes as these little dots. And the sperm head with its tail is shown. True, false? The sperm head has contacted the oocyte membrane. True or false? The sperm head has contacted the oocyte membrane. That's a true statement. So you have to be able to know what that is, right? What is the oocyte membrane? So that's kind of why I like to ask that question. Well, anyways, step 2, you can kind of see here that the corona radiata, it's still there, but it's kind of going away.
Is it all leaving because zona pellucida is now hard and pushing it off?
Yeah, that's a good way to think of it.
Okay. And where does that go?
The cells?
Yeah.
They're just cells, they just disintegrate and
Okay.
taken up by the tissue that surrounds it. I'll just say it's going away, but the ZP is still there. And keep track of the polar bodies. You should see the second one now because meiosis 2 completed. You ejected the second one. Well, I see 3. Now the 2 smaller ones are from the first one divided, but that's the second one right there. So a total of 3 polar bodies. They have like 2 kind of small ones. I'll just draw the polar bodies. But those are from the first. I'll just say from first PB, polar body. Then they draw a larger one. That's from the second. That is the second polar body. Okay, so they draw that there. But what's more important is what's happening on the inside. They call these pronuclei. Okay. So male and female. Pronuclei. They kind of swell. They both contain the genetic material. They always show these like these little structures there. I think that will become the spindle apparatus. And so here's our figure in the book, and here's the model we have in the classroom. It's over there on the shelf. So they do show a polar body there, but that's like the first one that's divided. The second one will come when those are ejected. So it's not exactly the same as this, but it's the close approximation. And here's the sperm. It kind of looks like a little parachute. I think those 2 red dots are supposed to be these 2 things, which will become the spindle apparatus. So it's a little off from the figure book, but that's what we have in the classroom. So again, this belongs to the father, male, female genetic material. So in the third step from the book, you have the DNA and the pronuclei. They're going to approach each other. And you start to get this mitotic spindle between them. Corona radiata is gone. The zona pellucida is still there. Pronuclei swell and approach each other. Remember, I'm calling this N plus N because you have 2 structures and they haven't intermixed yet. I see zona pellucida, there's no corona radiata, so ZP, check, and I see both polar bodies, okay? First, second, PBs, I mean, they're still there. Okay, just keeping track of it all. And you're starting to see that spindle that's important here. And we have a lot of models that show this. Here's 2 of them. That's the second one, that's the first one that's divided into 2. That's why they show kind of the 2 dots and 4 dots to illustrate that. But anyways, I kind of know that this one, and don't let the color code fool you. They call this male, although pinkish usually symbolizes female. But that's the male, that's the female DNA there. I mean, if you follow it backwards, this model, see the color there? And then see this color, this reddish color, so they kind of mean that this model, in their sense, then becomes this. This is still the male, and that's still the female. Okay, once again, that's the male, that's the female. So when they get to here, that's the male, that's the female. Then the chromosomes start to coil, supercoil. They condense. That's why this one comes out here. But all of these, in all three models, it's N plus N. The chromosomes have not intermixed yet. They're just kind of swelling and then they approach each other. So this is conception right here, zygote formation, the miracle of life. Pronuclei fuse. Chromosomes intermix. So you finally have what's called the N plus N becoming 2N. So it's like one pronuclei fuses with another. I'm not sure vertical. I usually show it vertically. So the chromosomes, they'll start to intermix. So that's just the pronuclei fusing. And call it 2N, diploid. And immediately it'll start to divide, because this is the one cell stage. Here's a real picture of it. The male and female pronuclei, they're kind of fusing together. You can still see the polar bodies there. So that's a real picture. And here's the model. I mean, call it the one cell stage, when it's all in one cell. One cell stage of development. This is pregnancy right here. Sperm fertilizes egg. It's one cell. You have to develop past this. So this is like day 14, ovulation and fertilization all occurs in the first day. So fertilization has been accomplished. Now you're going to start getting these cleavages of the conceptus of the one cell. So from zygote to blastocyst, here's what you got. It's undergoing mitosis now. We're done with meiosis, just regular cell division. So that's one cell. So 2 days post-fertilization, consider this day 1. I'm not going to write post-fertilization every time. Just the days now in this lecture are pretty much days post-fertilization. You start off with one cell. Cleavages are mitotic divisions where the daughter cells are like half the size. So, okay, pretend you got ZP, blue. It's still there. You have one cell. Okay, it's 2N, right? The one cell stage. A cleavage, you have to break up into cells that are half the size because you're contained by the restricted volume of the zona pellucida. So that one cell maybe becomes 2 cells that are half the size. And each of those are 2N, regular cell division. So one cell becomes 2, 2 cells become 4. Zona pellucida is still there. And they become half the size. They're all 2N, that's what I'm writing there. 4 cell stage, a couple of days post-fertilization. Then 4 becomes 8, and 8 becomes 16. So by around the 3rd day, you have a solid ball of cells, and they usually call that the morula. Days. I'm not going to try to draw it, but ZP, it's still there. This is 3 days post-fertilization. We're in the oviduct. Okay, I know it's not shown, but remember, the oviduct doesn't implant until around day 7 to 10-ish. Okay, so we're still in the oviduct. ZP is still there. But the cells that become a ball of cells called morula, if you look it up, it actually means little mulberry, basically 16 cell stage, a ball of cells. So there's no organization until you kind of get maybe a day later, day 4. We have what's called the early blastocyst. The blastocyst, the ZP is still present. I'll draw that as ZP. And inside, we see some organization. There's a little cavity, but there's these layer of cells on the outside. These cells that I've drawn in red, they're trophoblast cells. They are a supportive structure. It's not part of the baby. Okay? It's not baby. Supports baby. They usually call that extra embryonic. In its function, okay, quote unquote. That means not the baby supports baby. This actually will become part of the placenta. Okay. Now the inner mast cells that are stuck to one side there. Draw a bunch of cells. That's baby, okay? Those cells will differentiate into an embryo, into a human. Inner mass cell. That's future embryo. Embryonic. Not extra. It's embryonic. It's the baby. So that's what you got in day 4. Now, 7 days post-fertilization, they say it implants. Okay. Consists of a sphere of trophoblast cells and an eccentric cell cluster, like I already called the inner cell mass. So this one's ready to implant, day 7. Still blastocyst, but implanting blastocyst, same thing. This blastocyst has been allowed entry into the uterine cavity. And in the uterine cavity, there are lighted chemicals that finally dissolve ZP. It's finally gone. allowed entry into uterine cavity. Lytic chemicals there dissolve ZP. And once you do that, then now implantation is possible. Okay. So I'll say lytic chemicals dissolve ZP, making implantation possible. All right, so pre-embryonic development. Here's a picture of those stages in the oviduct and in the uterine cavity. I'll put a picture of the uterine cycle from yesterday's lecture right here, reminding us that ovulation must occur, usually does, around day 14 in a sexually mature female. There they show the fertilization by sperm. Okay, so call it fertilized, call it day 1, and then a couple days later, 4-cell stage. Solid ball, morula, 3 days, 4 days. So during the first week or so, there's a contraction at the isthmus. It kind of keeps the conceptus here, okay? Because you're not ready yet for the first week. You're not ready yet because you got to wait until you're well into secretory phase, right? So by design, keep it in the oviduct for a week, then allow it in, and allow it to implant at around day 7-ish, around there. So implantation of the blastocyst shown here, a little ball of cells. They cut everything open so you can kind of see inside. The little blue dot is supposed to be the inner mass of cells. And that's an ideal location for implantation, right there in the body of the uterus. So here it is if you don't cut it open. Basically what this is, around 7 days, 6-7 days after fertilization, it's just an adhesion. This is not implantation, but first you must stick. Adhesion. So it's still 7 days. Let's just say first step, adhesion of blastocyst to endometrium. First, adhesion of blastocyst to endometrium. So if you were to cut it open so you can see what's going on, it rolls, it sticks, and wherever it sticks, there's this kind of free rotation of the inner mass cells so that it faces the endometrium. Free rotation of inner mass cells face endometrium. Because now you're going to implant, and you want the inner mass cells to get in first through the door. So day later, 8 days post-fertilization, I'll call this invasion. This is the true implantation. Just digging a hole in there. So think of implantation as a couple of stages. Stage 1, adhesion. Stage 2, invasion. Eight days. Implantation. Invasion. you're going to see some different structures there. You have what's called the cytotrophoblast as well as the syncytiotrophoblast. Cytotrophoblast. Remember, trophoblast, not the baby. Supports the baby. Cytotrophoblast. Think of this trophoblast as, notice the cells are maintaining their cellular boundaries. So this is like just maintaining cellular boundaries around the baby cells. It's also maintains a cellular boundary. But the cytotrophoblast colored in yellow, I don't know if I want to use yellow. How about I use green? Kind of like this giant blob. And there's cell nuclei in there, but the cells, it's just, there's no cellular membrane integrity. It's all gone. It's all one fused cytoplasm. It's one big fused tissue. And they call that the syncytiotrophoblast. It's like a giant blob, and it's eating its way into the endometrium. That's how you're burrowing in, like a groundhog. Okay? That's the syncytiotrophoblast. Syncytiotrophoblast. It's doing the invasion of this step. It's secreting all these lytic chemicals. So I just say chemicals digest endometrium. I mean, you could look up the different chemicals. Like there's one called tumor necrosis factor alpha. You see HCG here in the syncytiotrophoblast being secreted there. You know what that is, right? Human chorionic gonadotropin. It has a function here, helps digestion, invasion, but as you well know, it gets into the bloodstream and it sustains the corpus luteum. Okay? That's there. So it's doing the invasion. It's getting in there. Now look at the organization that the inner cell mass takes. It organizes into 2 layers of cells. One layer. That one layer, call it a top layer, epiblast. Epiblast. Sometimes they just call it a superficial layer of cells. So epiblast, this is the baby. Or superficial layer. And notice a layer below it, colored yellow. That's the hypoblast. So they use yellow. I have yellow. Let's see if it works here. I'll color a little yellow there. called the hypoblast. Blast, when used to describe a cell, it's an immature cell. It's like it's not fully differentiated. Okay, it can divide and become something else. Well, anyway, it's called the hypoblast or deep layer. Most books are calling it epiblast, hypoblast. So the organization of the inner mast cells is basically now organized into two layers called the embryonic, bilaminar embryonic disc. That's on the slide there. Bilaminar means, bi means two layers. Embryonic disc. So call that bilaminar embryonic disc. We have some interesting things. This is the baby. However, there are cells that come from these blast layers. Like, for example, they show cells coming off the hypoblast that will actually become extra embryonic things. There are cells that come off of the epiblast. Okay, some cells are growing around here. If you kind of draw a layer of cells here, blue because they're derived from epiblast. They form a little cavity there, this blue space. You know, that space will become the amnion, the amniotic cavity, which will completely envelop the baby in fluid. Now it's just a little bubble. Okay? So that will become amniotic cavity. I'll just call amnion, same thing. And you see some cells coming off the hypoblast. They're developing this way. They're going that way in their development. Those are extra embryonic cells. They'll form an entire sac called the yolk sac. Extra embryonic just for support. So there's yellow cells forming yolk sac. You'll get a lot of stuff there. That's kind of the spot for the genesis of blood cells and blood vessels. It's not fully formed yet on day 8. So what I did was I rotated that picture like this, day 8, to make it look like that model, which is about the same time. Model's right here. Seven days. So this model is 7 days. Yeah, so we're about a day later. You can see the same things. Like, for example, I drew my syncytio trophoblast in green. I love that one for this model. For this model, I like to ask what this is. What do you think it is? Syncytio trophoblast. I like to ask that for this one. Now I see some similarities in the embryonic disc there. Superficial layer, deep layer. Superficial layer, deep layer. So that little bubble there is the amnion. You can see the yolk sac forming. Okay, so above there, that's just going to be endometrium, right? The endometrium. Now the goal of this implantation is to fully get inside, okay, under the blankets, right? You just get in all the way. The goal is to dig deep enough to nick a blood vessel. Because right now, all these cells are holding their breath. There's nothing to nourish them. There's no circulatory system. It needs to be bathed by mother's blood. So as long as you dig deep enough so you can nick a blood vessel and become bathed in maternal blood, then you'll survive. Well, anyway, it's day 7, day 8, around there. Keep on going. Now we're all the way in, day 9. Okay. So we're fully implanted. And I'll just kind of add what's new here. And distinguish between syncytiotrophoblasts versus cytotrophoblasts from that previous one. Nine days, what do we see? Okay. I see the bilaminar embryonic disc, just like before. I see amnion, I see yolk sac. Yolk sac fully formed. The cells of the yolk sac were derived from hypoblast. There's amnion, just like, it hasn't changed much. Okay, which trophoblast is this in pink? Cyto. The blob is syncytiotrophoblast. What's new here is we got some lacuna. Yeah. It's like, you know, when you dig a swimming pool in your backyard and you just kind of get the cement poured in there. But before you fill it with water, just space is lacuna. We're going to fill it with maternal blood. So add that. You get these spaces called lacuna at this day. That's new. Lacuna is singular. Add an E that pluralizes it. Will fill with maternal blood. And make a blood vessel. So they show the blood vessels, they show the uterine glands, which will supply glycogen. What we talked about it earlier. Now that you're fully in, we can now define what are called the 3 deciduas. I use this figure to kind of show it. Well, decidua is a word to describe the endometrium of pregnancy. Now, there's some changes that occur. The details you don't really have to know. Just know that the decidua is the endometrium of pregnancy. If we're successful here, we're not going to have menses, right? So this endometrium will persist for the entire gestation. And the decidua capsularis is the endometrium that overlies, overlies the structure, the embryo. Overlies embryo. The decidua basalis is pointing to decidua underneath. It's under it. Underneath developing embryo. Put a star next to that. It's really important. This is the maternal portion of the placenta. Super important. I'll come up again, and that's one student struggle with, identifying it. Okay, well, the rest of it, I mean, if it's not the part that overlies it or is under it, I mean, the rest of it that lines the uterine cavity is simply called decidua parietalis. Lines, rest, uterine cavity. Now, it's kind of hard to appreciate that when it's just kind of barely implanted. So I'm going to fast forward and skip back to here just to show you the 3 deciduas in a fully developed few-month-year-old fetus here. Hold on. Like, for example, when we get to this picture, a few months, 13 weeks, let's look at the 3 deciduas here. See this white-pink layer there? That's underneath, same size as the umbilical cord. That's decidua physalis. I said that's the maternal portion of the placenta because this is where the exchange is happening, right in there. Same size as the umbilical cord. So the blood flow is being exchanged. We'll go over that. But anyway, decidua physalis. Look at this light membrane on the other side. That's decidua capsularis. You can see that the uterine cavity is a sliver, but that the endometrium, or the decidua that's lining it, is the decidua parietalis. Okay. So those are the 3 deciduas. Let's go back to where we were. There. All right. So moving on. 12 days now. What do we got? So this is 12 days fertilization. What day in the ovarian cycle? What's 12 plus 14? 26. So maybe the female doesn't know this is happening. She hasn't missed her period yet. This is all occurring 12 days post-fertilization. And if you suspect you're late, you suspect HCG has to be in circulation, but it's not yet. Will be now, because now you've nicked a blood vessel. They say maternal sinusoid. A sinusoid is a blood vessel. Okay. You can see there, you're filling with the uterine glands with glycogen and blood. You're going to fill all those lacuna with blood. So that's new. Fill lacuna with maternal blood. Yeah, maternal blood, period. And you see the development of another extra embryonic tissue. It's called a mesoderm. So let's note that. It's colored in red in the figure. It's surrounding there. That's important. We'll look at it later as part of the placenta. So you now see extra embryonic mesoderm. Remember, it's extra embryonic, technically not part of the baby. It will support it though. All right, so an important event in early embryonic development is gastrulation, where 2 layers become 3. We had a bilaminar disc. It will now become 3 layers. And the endoderm, mesoderm, and ectoderm are their names of the tri-layered one. And they basically form all of the organ systems. So 3 germ layers that supply cells and development for organogenesis for all the body systems. And I list just a few here. I'm more interested in you understanding the process of how 2 layers become 3 layers. So once again, around 14, 15 days, it's 2 layers. Okay, so when I say 2 layers, look right there. It's still bilaminar. 14 days later. So let's kind of look at, this is a good figure. They kind of show you at around days 14 and 16, gastrulation. bilaminar embryonic disc becomes a trilaminar embryonic disc. So this figure, they emphasize, what I've been calling the two balloons. Well, one balloon is the amnion. The other is the yolk sac. Now the amnion and yolk sac are extra embryonic. The amnion provides a fluid-filled environment for the baby, although not yet. Provides a fluid-filled environment. The yolk sac is for the genesis of blood cells and blood vessels. It also helps form the mucosa of the gut, call it primitive gut. But the baby is the bilaminar disc right there. Okay, that's the two layers, right? Epiblast and hypoblast. So what they do is they get rid of the balloons on the figure, which I drew here. They cut it away, they cut it like that, and they cut that right down so we can see a sectional view of the two layers and how it becomes three. So what's going to happen is on around days 14 to 15, we get the first part of gastrulation. What's going to happen is you're going to have a migration of cells into this primitive streak, which is the beginnings of the vertebral cavity for the central nervous system. Cells will migrate from the top layer. Epiblast cells migrate into there, and they kind of dive into the streak to go to the bottom layer there. Migration of epiblast cells into the primitive streak. Now those cells that go in there, that first wave of migrating cells, they'll dive in there and they become the endoderm. The endoderm. This first wave of cells. So call it first wave. So these cells become the endoderm. Well then, day 16, post-fertilization, you get the second wave. Same thing. It's still a migration of cells, of epiblast cells, into the primitive streak. Second wave. Migration of epiblast cells into primitive streak. Those cells will become, they're colored red there, they're sandwiched in between. That's the mesoderm. The cells that remain, that did not migrate, you now call the ectoderm. Remaining cells, remaining epiblast cells, I should say, basically they did not migrate. They form that last layer, the ectoderm. So those layers, endoderm, mesoderm, and ectoderm, those are going to be the layers of your trilaminar embryonic disc that form the organogenesis for everything. Can you see them here? It's starting to form. That's day 16, gastrulation. In this section over here, they put a little lasso around the entire trilaminar embryonic disc. That's all I want you to see there. So, I've defined yolk sac and amnion. Let me define these other two. These are all extraembryonic supportive structures. The allantois, move back to a previous picture. Maybe it's on this picture here. Okay. This picture, you see a little evagination of the yolk sac. That becomes allantois. It will help form part of the umbilical cord. It also helps form urinary bladder. Right now you just see it as a little evagination, it's a little outpouching of the yolk sac there. But you can see how it's going to be the umbilical cord. I think that's clear. The other one was the chorion. Let's see the picture here. Ah, here it is. The chorion, the term is right there, and they put it around all of these three things. The chorion is pretty much the embryo part of the placenta. You have a mom part and a baby part, and they have to interact. The chorion is the baby part, decidua basalis was the mom part. Okay, this is a good spot for a break. Come back at 9:45 and we'll finish this.